Biology is all about comparisons, isn't it? And bioinformatics is especially that. And as the data grows and grows, there are more and more comparisons you can make. You can look at large comparisons, like comparing whole genomes to each other, maybe looking for inversions or rearrangements in genomes. And BV-BRC has a good tool for that, the genome alignment tool, which you can look across all the bacterial genomes and look for those kind of things. And let's say you're not interested in something that big, but you've got your particular gene and you want to find the close homologs to it. Wow. Every biologist knows about BLAST, don't we? And we use it all the time to find close relatives to our genes. But let's say you don't want to be as small as the individual gene, and you don't want to be as big as the whole genome. Let's say you want to be somewhere in between, a more narrow part of the genome. Let's say you're looking at maybe lateral transfer, or you're looking at changes in an operon. How can you look at that in BV-BRC? Well, there are some tools you could use, like the proteome comparison tool, which is a bidirectional BLAST analysis of up to 10 genomes, where you could look at the BLAST hits across all the genes. But still, let's go a little bit narrower. Well, BV-BRC has a great tool for that, and it's an old tool, as old as the BRCs. And it started here with the National Microbial Pathogen Database Resource, NMPDR. In the very beginning of the BRCs, there were 8 BRCs, and NMPDR was one, and Patrick was also one. And these guys had some powerful tools, and one was to look at gene neighborhoods. And if you look into this publication, you can see that they describe this tool, which they called the Compare Region Viewer, that starts with an initial gene that you see there in red, and then the gene neighborhood. And the same colored genes belong to the same protein family. So when NMPDR was wrapped into Patrick, and Patrick was wrapped into BV-BRC, this tool came along with it. So if you've watched any of these videos, you know that the way to find these tools is to click on the Tools and Services tab. And I can see BLAST, and I can see genome alignment, and I can see proteome comparison. But no matter where I look, I don't see Compare Region Viewer. How do I find that? Well, to find it, you have to go to the feature page, which is available for every bacterial gene in the genome, and you can look from there at the Compare Region Viewer. I've just put in a feature ID, and I'm going to click on the search icon. When it's searching, it's going to search across the whole database and give us the hits for the text that I put in. And right here under features, you can see this is a class A beta-lactamase gene. So let's click on this. Now, why this gene? This is part of our antimicrobial resistant workshop. So we're going to step through the same things that we would step through in a workshop with this gene. But look across these tabs, and for every protein encoding gene in the resource, you will find that it has this, a feature landing page that has similar tabs. And one of these is the Compare Region Viewer. So in the next videos, in order to do these comparisons, we're going to do a number of things. I'm going to show you how to create genome groups and feature groups. Then we're going to go to the Compare Region Viewer and manipulate it, because you can change how big the region is, how many genomes you want to see. All of those things we can do from this particular viewer. And then I'll show you how to export the view so you could use it in a publication. So I hope you'll join me. This is a really powerful tool, and it's a really great resource that we have in the system. Thanks.